And now we move to the research paper presentation, where distinguished researchers will share their studies on different particles and their effects on our health. And to start with the first paper, I would like to tell all of our guests that it's our practice to make our foods smell good, and in doing so, we usually find unique substances like the red gold of spices, commonly called as saffron or zafran. And it's not very easy to get it in its pure form. And how to authenticate that whether it's pure or not, because it's not only difficult to get it pure, but in the first place, it's not easily available also in the market. So we have our first presenter, Dr. Syed Rizwan, and the topic of his research paper is authentication of saffron, Crocus sativus, and detection of adulteration by using DNA barcoding. I would like to request him to please come here and make his presentation.
Assalamu alaikum. Good morning. I'm thankful to Hamdard Foundation give the opportunity to present at the occasion Health World Day, the theme Together for Health, Stand with Science. The topic today my presentation is authentication of saffron and detection adulteration using DNA barcoding. Agenda of my presentation today: introduction, background, study objective, barcoding methodology, results, summary of finding, and discussion. This slide is very important because it's a theme and vision of Hakim Muhammad Sayeed Shaheed. That is 1995, they established the Eastern Medicine Department or faculty in Hamdard University. And that project or this presentation is a collaborative research Hamdard University and Hamdard Laboratory. And I'm really feel too proud to say Hamdard University is the first university in the Eastern Medicine or TIP science. They're doing a work with molecular biology. As we know, saffron is a red gold, is a very expensive and precious herb used in spices as well as herbal medicine. Usually, we should talk before specific efficacy, and we should make sure identity of that particular herb or material that's used in the medicine. Because saffron is a very expensive and not cultivated everywhere of the world. So high value make to a high adulteration chance. As you know, the why we call the red gold, because 150,000 flower of stigma that give only 1 kg of saffron. It means this property to make a expensive and precious among the spices as well as among the herbs. This map indicates why the saffron is expensive, because it's not cultivated in all over the world. It requires specific weather and property of specific soil. That's why they are grown very specific, limited part of the world. Because adulteration lead to be the risk, either economic, either public health concern. Because if we use anything adulterant regarding especially saffron, either in spices or either in herbal medicine, that lead to be economic concern as well as public health. The two types mainly use adulteration in the saffron. One is whole and second is powder. What it means a whole? They use a whole stigma of the different similar flowers, and such as corn fiber, silk fiber, and etc. And similarly, they use also the powder such as turmeric, paprika, ground seed, fennel, gardenia extract, and artificial dyes such as Sudan and rhodonamine B. The traditional method, if we compare to the modern method, such as molecular biology, as name, such as DNA barcoding, is traditional method is fail some extent to by their visualization and microscopy, and some extent to chemical analysis. And modern method, why they are success? Because they detect at the molecular level, such as DNA, protein, and RNA and carbohydrates. The DNA barcode, with the help of that technique, we find the minor level of adulteration at the molecular level. Now, what is the DNA barcoding? For the common understanding, when we went to supermarkets, at the time of paying, cashier barcode the things. If you observe, the different barcodes have a different price. Similarly, nature gave each human being have a separate barcode in the form of DNA hereditary material. That material is specific to species. Okay. How they work? Usually DNA barcoding is a barcode, gene marker. If you, for the understanding, the people, kasoti. The gene marker is a kasoti to specify or recognize the species specific to species plant. That's why RBCL is our gene marker or kasoti to identify with the help of DNA polymerization process to find out the specific species or not, if we find some alteration in our sample or any required matter. That is a diagrammatic presentation. First, usually we get, if we need the DNA, we collect the sample and extract the DNA required following sample. After that, when we extract the DNA, then we amplify the DNA from the sample and our gene marker, so we call it kasoti. After amplification, when we get the amplification product, that product usually we send to Korea, well-known company for sequencing, Macrogene. That when it's sequence, then we receive the sequence from them. After that, we analyze with the help of bioinformatics tool, such as BLAST. With the help of BLAST, we can predict the results and final conclude either this product is adulterant or not. Attractive study, evaluate an authenticity and safety of market saffron using DNA barcoding. Because without authenticity and purity, we can't use anything either in herbal or spices to make sure the authenticity to use before any product. The methodology, as explained, when we collect the sample, we grind and purify with the help of specific biological protocol to extract the DNA. That DNA, then we also use the protocol for PCR, stand for polymerization chain reaction. After that, that product usually sent to for sequencing to further analyze the results. This is diagram. If you see, this is diagram usually known as DNA gel electrophoresis picture. I use the word in the previous slide, the polymerization or amplification process. When our target gene to hit our DNA sample to get in the form of lights, bright bands. That band, it means our our product is pure and is not adulterant. This whole methodology, how many steps include the PCR product purification, DNA sequencing, sequence assembly, sequencing identification, and phylogenetic analysis. Okay, sequence identification, usually we use the BLAST tool. BLAST is a method in which we use two sequences. One is reference gene, they are present in the gene data bank, and another gene the use which are collect from our sample. If they hit 100% or more than 90%, then we conclude or say that this sample is pure. As you say, BLAST, one sequence is referencing, as another sequence is subject. If you see, the whole sequence is matched to each other. It means our sample is pure, is not adulterant. Because we use 4 samples, different type of saffron collected from market, and we analyze the 4 our sample is pure, and sequence, if you see, are aligned and engaged. And these are the results. If you see 100%, 99, 100% and 99, means these all 4 samples are pure and is not is adulterant. As the results, saffron is pure. If we find any sequence adulterant, means the DNA sequence is not match 100%, maybe just match 40 or 50%. Okay, summary of finding. DNA barcoding successfully identified to authentic saffron or crocus sativas with 100% match. If, as I told you last slide, if any adulterant sequence or sample present, it means their sequence is not match 100% to our target gene. And one is very example I'm going to present for the public. If you see the black seed and the onion seed, the morphology of both seed are more than 90% is similar. Means if you want to see by the visualization, you can't identify that is one is black seed, another is onion seeds. That's why here is visualization and chemical method, they are failed. Then come to technique DNA barcoding, because the DNA reveals the truth, either I am pure or not. Discussion. DNA barcoding effectively authenticate crocus sativas and identify adulterant samples in the market supply. Molecular identification prove more accurate and reliable than traditional morphological method. As I gave the example last slide, morphological, onion seed and black seed. If you have another example, and many, there are resemblance, morphology more than 90%, but they are not actual same species. That's why DNA barcoding play a very important role, not only authenticate, but also play a help to make a good production, or to give to confidence in economical as well as health sectors. Thank you.
Thank you, Dr. Rizwan, for your very nice presentation, insightful presentation.